Alrighty, so yesterday I had to teach you guys this from home because internet business and all that fun stuff. So I'm re-recording this lesson for you guys just so that we have it because I wasn't able to record it at my house. So going through the process of transcription. Again, I want to just recap the whole replication process first. So DNA, as we've talked about, we have two backbones. The backbones are made up of sugars and phosphates. In DNA, it's made up specifically of deoxyribose sugar. And then the rungs of the ladder, or the inner parts of the DNA molecule, are those nucleic acids or nucleotides or bases, whatever you want to call them. And again, it's a double helix molecule. It's kind of like a spiral staircase. And during the process of cell division, we need to make sure that we are replicating this DNA so that we can have the exact same copies in both of our new daughter cells at the end of this replication process. So how this replication process happens, again, we have kind of 6 general steps, and those 6 general steps stay the same. They happen in the same order. They don't change on you. So first step, you have the helix. We need to unwind it. So DNA helicase unzips the genes or unzips the DNA. Then we have those single-stranded binding proteins attaching to those open strands to make sure that they don't just bind back together. So DNA helicase opens the DNA and then the proteins hold it open. Then we have a primase enzyme come in and attach primers to the DNA molecules so that we have a starting point, basically. And then DNA polymerase 3 is going to attach to these primers and then start to just add on some nucleotides to our DNA molecule. So what that looks like, I'm going to try and zoom in a little bit more, if I can, on my board up here for you. Excuse my awkward techie skills. Here we go. All right, so if my DNA molecule was this, okay, my primer, let's say, will be this. And that is my start point for my primase molecule. And I know this is kind of like backwards, so I apologize for that. I don't know how to mirror this image on you. When DNA polymerase 3 comes into play, it's going to add DNA nucleotides. And again, these nucleotides are going to be complementary to the DNA strand. So A's are going to bind with T's, T's are going to bind with A's, and G's are going to bind with C's, so on and so forth. All right? So helicase opens it, single-stranded binding proteins keep it open, and then we have our primase attach a primer, and then DNA polymerase is going to add nucleotides. This one we would consider, again, the leading strand, okay? Because it is created continuously, which means that DNA polymerase 3 attaches one time and then just zooms down from the 5' to 3' direction. On the opposite side, so our lagging strand, apologize, I wrote my primer wrong up here. Okay, this one's our lagging strand. Our lagging strand, again, is going to be a little bit wonkier because it is kind of being synthesized in a discontinuous manner. So we would have to have our primer at a couple different start points as the DNA unwinds. So let's say we have a primer added here, and we have a primer added here, just to pick 2 spots. So DNA polymerase first would synthesize this DNA. Okay, so it would attach to this primer and then work from the 5' to 3' direction. And then it would attach to this primer and then fill in the rest of this DNA. It's early, my brain isn't quite here yet. There we go. All right. So the helix has been opened using helicase, sorry. The single-stranded binding proteins have bound to keep these strands open. Primase has added the primers, and DNA polymerase 3 has added the nucleotides. Okay, the next step is that DNA polymerase 1 needs to come in, and it's going to replace our primers with DNA nucleotides. Okay, so it does that on the leading strand. Then it's going to come down here to the lagging strand and do the same thing. So that we have 2 identical copies of our DNA molecule. And then the very last thing that needs to happen is DNA ligase comes in and it just joins together that sugar phosphate backbone to make sure we have a nice, solid, full molecule. So again, helicase opens the genes, proteins bind to keep it open, primase attaches RNA primers, DNA polymerase 3 adds DNA nucleotides, DNA polymerase 1 changes out your RNA for DNA, and it's also going to do a quick little kind of spell check to make sure that all of the bases are paired to the right thing, that you have T's with A's and G's with C's. And then ligase, again, lastly, is going to join the backbones together to make us a nice, happy, complete DNA molecule. Woo, sorry. I'm going to zoom back out here. And bring us back to the screen. All right. So DNA, again, is semi-conservative replication, so that means that each new DNA strand is going to have an old strand, so I have those written in black on my board, and then the new strands are the blue colors that I wrote throughout the replication process. Okay, and then I did this example with you guys. It is on the PowerPoint. Again, the blue strand is my template strand, and then my pink strand is my newly synthesized or newly made strand of DNA. So how do genes in DNA determine an inherited trait? Again, I showed you guys this video about what a gene actually is. And genes, again, are just small parts of DNA. So when we are doing gene expression, so when we're looking at taking your hair color and actually expressing that hair color, so showing that you have brown hair, or showing that you have blonde hair or red hair, or whatever kind of hair you have. To express those genes, you are not going to be transcribing or turning your entire DNA molecule, the entire 2 meters worth of DNA, into proteins. Okay, we are taking small portions, so maybe it's only 20 base pairs long, so 20 bases of DNA. Maybe it's 100 bases of DNA long. Maybe it's 1,000 bases of DNA long. Okay, our DNA again has over 4 billion base pairs, which means that we have a lot of genes crammed into it. But when we're expressing a specific gene, we are transcribing, which is the process I'm going to talk about in a second, just small parts of the DNA. And all of your genes can be expressed through the process of making proteins. So DNA is always going to code for proteins. Proteins are the physical showcase of your genetics. Okay, and proteins start out, they have primary structures, secondary structures, and tertiary structures, which we generally talk about in Bio 20, but again, with, online COVID business last year, I don't know. But primary structure is just a chain of amino acids or a polypeptide chain. That is what we get to at the end of this protein synthesis process. And then that polypeptide chain can then go off to wherever it's going to go, and it's going to fold into the 3D functional protein like hemoglobin or keratin or whatever other protein we may use, like different types of enzymes. Okay, so genes, every single gene in your DNA will eventually make a protein. We also have a different structure that we're going to make through this whole transcription process, and it's called RNA. So DNA, again, is a double helix structure. Sorry, I'm taking forever to erase my board. So you have one backbone, and then you have a second backbone. And it's a helix because it's kind of twisty. And then you have your nitrogenous bases, your nucleotides, whatever you want to call them, in the middle. And again, these are A's, T's, G's, or C's. The sugar that we have, because again, you have a sugar hooked up to a phosphate, and the sugar is deoxyribose. Okay, whereas in RNA, it also has a sugar phosphate backbone, but the sugar that it uses is ribose. Instead of thymine, RNA uses uracil, so it's going to have a U binding with A instead. And it is a single-stranded molecule versus this double-stranded molecule. It is still kind of helical, so it can still kind of wind up on itself. There are points where your RNA molecule can actually bind with itself as well, so these bases can pair. So you can have some double-stranded RNA, but it is a single molecule that, again, those bonds are very easily broken, and it turns back into a single-stranded structure. So the sugar phosphate here, this is a ribose sugar, and again, the bases are going to be A, U, G, and C, versus the T that we have over here, okay? And so this picture showcases it a little bit nicer than what I can draw. So again, DNA is double-stranded, RNA is single-stranded, and we have a U in place of the T. And then for the sugars, again, the big difference is the fact that ribose has an extra oxygen at this specific point in the molecule, which is why we call DNA deoxyribose, because it's deoxygenated at one point. We also have 3 different types of RNA. So messenger RNA, this is the one that we're going to be looking at for transcription. Okay, so this is the one we're going to be talking about today, because this has to do with transcription. Transfer RNA is what we're going to talk about later today, okay, this afternoon. This one has to do more with translation. So taking your mRNA molecule and actually physically turning it into your amino acid chain. And then ribosomal RNA is the RNA that physically makes up the ribosome. Okay, so the ribosomes, if you remember back to Science 10 in the parts of the cell, ribosomes are just a bunch of those little dots that are floating around in the cytoplasm, pumping up proteins, because that's their job. The ribosome, you also have them on the rough endoplasmic reticulum, so that weird kind of swirly stuff hanging out by the nucleus. And again, those ribosomes that are sitting on the rough ER are pumping up more proteins. And then the ribosomes themselves are made inside the nucleus, specifically in the nucleolus, okay, where the DNA isn't part of. So the nucleolus is making these protein factory functional things, okay? So transfer RNA and ribosomal RNA, these ones are both involved in the making of proteins, which again is the process of translation. All right. So the central dogma of molecular biology, which is what we're talking about now, is the fact that DNA makes more DNA through replication, then DNA makes RNA through transcription, and then RNA is used to make proteins through the process of translation. This always happens. It doesn't change. Again, it's the same order. You replicate your DNA, then you can transcribe your DNA, and then you can translate your DNA. So basically, DNA codes for proteins is what you guys need to remember. Okay, DNA again makes RNA through transcription, and RNA makes proteins through translation. So the transcription process itself, this is where we're taking DNA and turning it into RNA. And when we relate it back to DNA replication, the process itself is a little bit more simplified. Okay, it's not as many steps. It's not going to be as necessarily complicated for our purposes. If you take this in university, it does get more complicated, but for Bio 30 level, it's not as bad. But we have our DNA molecule, which is this blue strand here, and the DNA molecule has to open, so it gets unwound for the process of transcription. And we use one of the strands, typically the bottom strand, in a lot of the pictures that you'll see, as a template to, again, create the mRNA strand, which is this red one here. And the enzyme that we use in this case is RNA polymerase. So again, anytime you hear the word polymerase, it just means to add nucleotides. DNA polymerase 3 adds nucleotides during replication. DNA polymerase 1 changes RNA nucleotides for DNA nucleotides. RNA polymerase, which is the enzyme we're going to use here, adds RNA nucleotides. Okay, so polymerase means adding nucleotides, building a molecule. When we get into biotechnology next week and we start talking about the PCR reaction, so polymerase chain reaction, again, polymerase adding nucleotides in a chain reaction. And I'll be able to talk a little bit more about COVID testing at that point because the test that they are using for COVID right now is a PCR test. So it's being used in action all the time. I lost my pen. All right, so there are 3 steps to the transcription process. We have initiation, aka it starts. We have elongation, or the mRNA strand gets longer. And we have termination, or the entire process stops. It's pretty self-explanatory. Fun fact, the stages of translation that we'll talk about later this afternoon are the same, which is awesome. There's just different things that happen. So during the initiation process, and I'm going to just, again, move you over to my board over here. Zoom in. All right, and then I'll go back to my other board to show you the fancier diagrams than what I can draw. So with initiation, this again is the start, okay? When we're starting transcription, we basically need a starting point for this RNA polymerase molecule to actually attach. So what happens is your RNA polymerase, and I'm changing it just to POL instead of writing out polymerase every time because it just gets to be a lot. So RNA polymerase attaches to the DNA. At a promoter sequence. And that promoter sequence, or that start sequence, we call the data box. And the reason we call it a data box is because it's an area of DNA that has a large amount of A's and T's. It does not necessarily have to be in the order TATA, but it's just a large section of DNA that's like, I don't know, 10 to 15 base pairs long, potentially, of just A's and T's very much put together with no G's or C's in sight. And so RNA polymerase kind of sees this as a giant neon sign saying, please start here, and so it'll attach. So if we have our DNA molecule, and you have, again, a bunch of A's and T's. I did not draw this molecule big enough to do this. Okay, we would refer to this again here as the promoter sequence, and then our RNA polymerase can attach, and the process can start. Alrighty. Then we have the elongation phase, which again is just building the actual mRNA molecule. So your RNA polymerase moves down the DNA. Again, in the 5' to 3' direction. Okay, and it's going to unwind it, unwinding it. And then adding nucleotides. To the growing mRNA molecule. And the DNA understands as well, as soon as the RNA polymerase moves down, it's unwinding as it's going, and then it rewinds right behind it. Because again, those bases in the DNA molecule are very much attracted to each other, and they want to stay bound, okay? So if we look at it kind of like this, we have some unwound DNA. And I'm only going to write the nucleotides on the bottom, just because it's easier. And then our RNA polymerase is attached, and it's moving this way. And then we're going to add some RNA that again is going to be complementary. So the bases will pair for the process of making it, and then they'll move off. And I can zoom in a little bit more to this specific area here in a second. The big thing that will be different, again, because it's an mRNA molecule, is that instead of adding T's, you need to add U's. So I'm just going to zoom into that one specific spot right here, just so you can see it a little clearer. I was really bad at first-person shooter games, which is why I'm struggling. So again, as you can see, I have U's bound with A's. Okay, A still goes with T, C goes with G, G goes with C, but again, the U is binding with the A instead. And then for termination, our last stage here is when we need to stop this process. So as we get to the end of this gene, because again, transcription does not happen for the entire DNA molecule. We're not doing all 4 billion bases, because then that would just end up with something weird at the end. We just want to have a specific gene to create a specific protein to express a specific trait. So when we get to the end of that gene, the RNA polymerase hits a termination sequence. And in this case, it's typically going to be a string of A's. So you just have a bunch of A's in a row. So 10, 15, 20, 50, whatever, just all A's. And the RNA polymerase is like, okay, I'm adding so many U's, like this is ridiculous. And then it just stops because it's like, okay, we're done. That's enough U's. We're finished, okay? So after it hits the termination sequence, RNA polymerase is going to release the DNA. It says, okay, see you later, DNA. I'll come back and transcribe you another time. And the mRNA, and then the mRNA needs to actually go into the cytoplasm. So the mRNA is leaving the nest. It's going on to bigger and better things. It enters the cytoplasm, which again is where we have all of these ribosomes hanging out, because the ribosomes are really important for translation to turn this mRNA into an actual protein. So it's leaving our nucleus, going on to do its thing, and then RNA polymerase can then find another part of DNA or another gene and start to transcribe that and just basically make a whole bunch of mRNA molecules forever and ever and ever. So what this looks like, again, you have your DNA left behind. Pretending that it's helical, I should actually draw it like that. Oh goodness. Okay, your mRNA is going to float off to do its thing. And then your RNA polymerase is ready to again find another gene, transcribe it, and do its transcription thingy. All right, so coming back to my other screen here, one day I'll give it first try. One day it'll happen. Today is not that day. All right, so initiation again, RNA binds to that promoter sequence or that TATA box to get this party started. So the promoter again is just strings of A's and T's. It's an initiation sequence, a promoter sequence, whatever you want to call it. Looks something like this. So we have our DNA transcription unit. Again, is just the gene that we're looking at, so not the whole DNA process. The promoter is just this short sequence here. RNA polymerase attaches. We're good to go. Elongation. Again, RNA polymerase just books it down, the DNA gene from the 5' to 3' direction, adding on RNA nucleotides to that growing mRNA strand. Again, remembering that U replaces T in RNA. Then we get to the end of that process, the end of the gene. We get to this string of A's, which again is that termination sequence saying, okay, RNA polymerase, this gene is done, go forth, find another gene, we are done. So it's going to release the DNA, it's going to release the mRNA, and then the polymerase is going to go and find a different gene. This process, again, is going to create this little hairpin loop structure where your RNA is going to bind with itself for a couple minutes, and then it will become single-stranded again a little bit later. And the mRNA, again, is going to travel into the cytoplasm, where it can eventually meet up with a wonderful ribosome and turn into a protein. And then these are just some summary slides for you guys if you want to go back to it. Awesome. And then the other example that I gave you guys was replicating a strand of DNA. So the blue strand, again, is the original DNA, or our template strand. And then this pinky purple kind of color is the replicated DNA strand. I used the same template strand on the bottom here to transcribe, just so that you guys could see kind of the similarities and differences between the two different strands. So again, G is going to bind with C. It kind of like messed up my order a little bit, so I do apologize for that. U goes with A, okay, A goes with U, T goes with A. So to draw that over here, I just want to take a picture of this board for you guys quickly. And then I will draw this out real fast. Just so that you can see it where it's actually matching, because this is driving me nuts, how it's not actually matched up. Okay. So if my original DNA was this, okay, if that's my DNA strand, my RNA strand that I would make from that, okay, would be A, because T still binds with A's. Then I would have a U, because A now binds with U. A, C, G, C, A, U. Nope. U, A, 5'. Okay, eventually, you guys will get this process. I did give you this practice sheet to do yesterday. So please make sure you do that. And then this afternoon, we're going to get into translation. So again, we're going to be able to take this mRNA molecule that we just made. Okay, we're going to rewrite it down here. Just to give you a quick little view. Again, we want to always write it from the 5' to 3' direction. So I literally just took this and I flipped it around so that it will be in the way that we'll actually be reading it. And with translation, what we do is we take this mRNA molecule now, and we're going to split it into groups of 3, and then the ribosome is actually going to read these groups of 3 and create your polypeptide protein chain based on these 3 little segments. So that's what we're going to get into later. So hopefully this is helpful for you guys.